As you know, hepatic encephalopathy is a common complication of cirrhosis, and its pathogenesis is related to the effect of the ammonia in the brain and can be exacerbated by inflammatory mediators. Classically, hepatic encephalopathy is considered as a reversible pathology, and the alterations reverse after liver transplantation. However, recently one finding in our lab describes some brain damages in patients that makes persistent cognitive impairment. The patients presented some brain atrophy, and this cognitive impairment seems to be related with the number of episodes of HE. So our hypothesis is that the different episodes of HE and that inflammatory processes causes a dysfunction of astrocytes, microglia activation, all that can lead to neurodegeneration. Our objectives here is to identify the elements that contribute to neurodegeneration process in a new experimental model of episodic HE, and to assess the relationship between nitrogen metabolism, inflammation, and neurodegeneration by means of a differential analysis expression. To get our results, we developed a new animal model using rats. These rats were infused every 2 weeks during 5 months, trying to simulate the different episodes of HE that present the patient of cirrhosis. We had one sham group that were infused with saline solution, and we had 4 portocabal anastomosis rats. That one group were infused with saline solution, another one with ammonia solution to simulate the hyperammonemic conditions, another one with lipopolysaccharide to simulate an inflammation, and the last group were injected with a mixed solution with ammonia and LPS. As we expected, at the end of the treatment, we observed an increase of ammonia blood levels in the ammonia group, an increase of pro-inflammatory cytokines in both LPS groups, and an interesting decrease of reflexes in the ammonia group. If someone wants to know more details about this model, you can take a look to the poster of my lab mate that is titled New Animal Model of Episodic Hepatic Encephalopathy. Here I'm going to present only the results of the differential expression in cortex and cerebellum, but in parallel we are also doing some histological studies in different brain regions to complete the results. Okay, for each animal we did 2 arrays, one for cortex, one for cerebellum. Each array was able to analyze 16,500 genes at the same time, and we included 5 animals per group, except the mixed treatment that we only could include 4 animals. Okay, array is a tool that generates a lot of information, as you know. It's important to say that we have all the comparisons of each group against the other groups, but we only exposed today each group against the sham group, because this is the point where we started to analyze all the data. Okay. To focus our search, we start to investigate the genes that have been directly related to be involved in nervous system. Okay, that's the marked genes. To put some light here, we use an Ingenuity pathway analysis software to get some connections between genes, and in cortex, under ammonia conditions, we found 2 interesting genes that are adenosine 2A receptor and dopamine receptor D2 that were involved in behavior alterations and some addictions, or related in some activities related to nervous system, as activation of neuroglia, synapses, neurotransmission, and so on. In cerebellum, we found different genes that are involved also in the nervous system, and using the generative pathway again. Here we found the highest overexpression in the experiment. This transthyretin gene, that is 17 times overexpression. Transthyretin codified for a carrier protein and is related to amyloidosis in humans. Using the generative pathway analysis software, we selected 2 genes more here, the transcription factor OTX2 and the serotonin receptor HTR2C that are related to movement disorders and related to formation or development of different brain regions. We didn't find any canonical pathway to englobe all the genes in some way to explain all the changes that we observed in the rats. And well, it's probably that in cortex as in cerebellum, the addition of the different effects of several genes causes or could cause some kind of degenerative processes. To confirm our array results, we did a real-time PCR with the first selected genes, and as you can see here, the numbers were very similar, so we can conclude that our array results were were reliable. To start in cortex, we checked ADORA2A, and it's clear the downregulation of this gene under ammonia conditions in comparison with the sham group, and that's validated with a significant decrease of that protein in a Western blot in the ammonia and portocabal group respect the sham rats. In the case of TTR, the transthyretin, that's the main overexpression. It's important to say that not all the rats presented that overexpression, but only 50% of our rats present a huge overexpression, around 200 times, and the other 50% of rats have an expression that's very similar to sham or even lower expression. We would like to show you an amazing picture of a Western blot, but that's not possible because we are having some troubles with the antibody trying to reproduce the results. But at first sight, that overexpression seems that it's not present at protein level. Okay? So in summary, in LPS groups, we showed an increase of inflammation-related genes as the complement C3, the chemokine CXCL13, and genes related to neuronal death via apoptosis as LCN2. LRG1 is also increased and is related to cause neurodegeneration in mouse models. And CCL2 in cerebellum is contribute to microglia activation also. In ammonia group, we observed downregulation genes, 2 genes interesting as ADORA2A and DRD2 in cortex. We saw an upregulation of transthyretin and HTR2C and OTX2 in cerebellum, that both are genes related to neurodegenerative and movement disorders. In conclusion, the results obtained under precipitant factor conditions of AG, like hyperammonemia and inflammation, show alterations in genes related to neurological and movement disorders. These alterations could lead to neurodegenerative processes that could explain the cognitive impairments in AG patients, and obviously further experiments are required to demonstrate that degenerative evidences. That's all. Thank you for your attention.